So last class you guys made candy DNA molecules, and we learned about basically the structure of DNA. So the shape, remember, is called a double helix. It's that twisted ladder shape. Inside your DNA molecule, like the rungs of the ladder or the steps of the ladder, were your 4 nitrogen bases, and they pair up. So that was your A's, T's, C's, and G's. And then they're connected to the backbone of your DNA molecule with hydrogen bonds. And then the backbone is made up of sugars and phosphates. And the hydrogen bonds connect specifically to the sugars. So in this lesson, what we're going to do is we're going to focus on kind of building up our entire DNA molecule, not just a couple base pairs, but thousands of nitrogen base pairs, and see what that makes. So first we're going to watch this video to refresh our memories about DNA and what it's made out of.
Reader beware, you're in for...
Geckos and grasshoppers, worms and watermelons, elephants and Escherichia coli, man and mushroom. All so different in form and function, but amazingly the same in how their form and function are determined. First, all these organisms are made of one or more cells, and every cell of every living thing on Earth contains all the information it takes to create and duplicate and make variations of itself. That information is stored in a very long but quite simple double molecule called DNA, or deoxyribonucleic acid. And the DNA of every living organism is made of chains of 4 smaller molecules called nucleotides. What dictates the difference between a man and a mushroom is the sequence of these nucleotides in the long DNA chain. The 4 differing nucleotide parts, called bases, are made of a few carbon, oxygen, hydrogen, nitrogen, and phosphorus atoms, and the molecules look like this. And each of these 4 bases is attached to an identical backbone molecule, a sugar called deoxyribose, the D in DNA, and a phosphate group. Let's simplify these nucleotides and show them like this. So, a single sequence of nucleotides joined by their common sugars would look like this. And the DNA molecule where such sequences are stored looks like this. But how does a simple molecule dictate the form and function of millions of different living things? You can think of DNA as a great library of information. Information that is used to do one thing and one thing only, direct the building of different protein molecules. And it's the proteins that build the cells and keep them functioning and changing and reproducing. Here's where the familiar word gene comes in. If your DNA is a library of information, a gene is a book in that library. A gene is a segment of the DNA molecule. Let's say your body needs a particular protein, like insulin. To get it, some of your cells send a protein signal through the bloodstream to the cells in your pancreas, where insulin is made. That signal protein tells other proteins in the cell's nuclei to open up a part of the DNA double helix, the insulin gene, and start making insulin proteins. As soon as enough insulin has been produced, another signal protein comes to the pancreas's cells that tells them to stop making insulin. It's like looking up a book in the DNA library about insulin and then putting it back when you're done. There are genes in DNA for visible and invisible things that make up your body, like genes for eye color protein pigments, for skin color, for hair color, for stopping and starting bone growth, for your blood type, for how many fingers or arms and legs you have, for proteins that influence how long you live. Your DNA probably contains between 25,000 and 40,000 genes, while the DNA of a worm or a plant or a fruit fly contains about 12,000 to 20,000 genes. Some of those genes have quite different sequences of nucleotides than yours, and some are similar to yours. Though it happens infrequently, our own nucleotide sequences can change as the result of spontaneous or environmental damage, which might remove or shift a nucleotide position. This changes the gene involved and can then change the protein. Most of these changes, called mutations, have very little effect on the organism or its descendants. Some are mildly damaging, and a few can make the organism better suited to its environment. It is these tiny changes in DNA gene sequences happening over millions of years that create the differences among living organisms, from geckos to grasshoppers, worms to watermelons, elephants to Escherichia coli, and man to mushroom.
So we had talked about this already during your lab, but the nitrogen bases, they pair up in a very specific way. So the A is always paired with the T, and the C is always paired with the G, or vice versa. T pairs with A, G pairs with C. So the way I always like to remember this is I use the mnemonic device, apples in the trees. So A and T together, and cars in the garage. C and G go together. We're going to come back and watch this video at the end. So I need you to follow along with your notes, just like we always do in class. So we have a lot of DNA in our bodies. So how does it all fit in to our nucleus? Remember, the nucleus is that central part of the cell. In grade 8 you learned that it's kind of like the brain of the cell. So DNA is packaged up. And it's packaged up into things called genes. Genes are special, uninterrupted segments or sequences of DNA. Now that word DNA can be kind of confusing sometimes. So DNA sometimes refers to the nitrogen bases, A, T, C, and G, or sometimes DNA also refers to your entire set of chromosomes, all 46 chromosomes. So here they're referring to DNA as your A's, T's, C's, and G's. So what a gene is it's segments of A's, T's, C's, and G's as they're partnered up together. Just like the video said, these genes provide the instructions for a lot of our traits. In section 2, we talked about different kinds of traits: discrete, continuous, heritable, and non-heritable traits. So remember, a trait is something that's kind of expressed in an organism, or it shows up in an organism, like your eye color or your hair color. Genes can be made out of hundreds or even sometimes thousands of pairs of nitrogen bases. So they're actually really long segments of nitrogen bases put together. Your genes come in pairs. So you get one set of genes from your mom and the other set from your dad. So you're inheriting one gene from each of your parents. And remember, that word inheriting means it's passed down from parent to offspring. So for example, if we look at the gene for eye color, you inherit one gene from your mom, and you also get one gene from your dad. Those genes, or how you get them from your mom and your dad, is those genes are found in either the egg from your mom or the sperm from your dad. Not all copies of the gene are identical. So obviously for eye color, there's a wide range of eye colors. So they're not all identical, otherwise we'd all have the exact same eye color. Alternate or different forms of a gene are called alleles. So again, going back to eye color, you can have a gene for eye color, and depending on what you get from your mom or your dad, you get different alleles. So alleles could be like blue eye color, brown eye color, green eye color. So alleles are different forms or different types of a gene. So let's go through an example a little bit in more detail. So you inherit your eye colors from your parents. It's a heritable trait. Eye color is encoded by one gene. In reality, it's actually way more complicated than that, but for now, we're just going to keep it simple. So we're going to say eye color is coded by one single gene. And remember, those genes are made up of a long sequence of A's, T's, C's, and G's. You receive one gene from your mom and one gene from your dad. So you get your gene from your mom from her egg, and you get the one gene from your dad from his sperm, and then remember they fertilize and create that very first cell called a zygote. Once you have a zygote, you have two copies of a gene, and each of those genes are alleles. So you get one allele from each of your parents that encode for the gene for eye color. The allele may or may not be identical. So one allele might code for green eyes, and the other allele from your other parent might code for blue eyes. Green eyes or blue eyes, those are different alleles of the gene for eye color. Which one shows up? So are you going to have green eyes or blue eyes even though you have one allele for each of those eye colors? Or we're going to talk about that later on in section 3 when we talk about patterns of inheritance. Next we have chromosomes. Chromosomes are packages of genes. So when we put a bunch of A's, T's, C's, and G's together, those nitrogen bases, we get something called a gene, and there's different forms of a gene called alleles. When you put a bunch of genes together, you get something called a chromosome. Chromosomes are arranged in pairs. And just like before, you get one of the pair of chromosomes, or one half of the chromosome, from your mom, and the other chromosome from your dad. Each chromosome contains one allele. So if we're talking about eye color again, you get one chromosome from your mom that contains an allele for eye color, and you get one chromosome from your dad, which also contains an allele for eye color. Each human body cell contains 46 chromosomes. So that means overall we have 23 pairs of chromosomes, 46 total chromosomes. This is only in body cells. So body cells are literally every single cell in your body except for an egg or sperm. Now, when I said you get half of your chromosomes from your mom, half of your chromosomes from your dad, what that means is that you inherit 23 chromosomes from each of your parents. And that's, again, contained in the egg and the sperm. When the egg and the sperm unite and they fertilize to make that very first cell called the zygote, then you have an entire set of chromosomes, because 23 chromosomes from the egg, 23 chromosomes from the sperm, you get 46 total chromosomes. Now, different organisms have a different number of chromosomes. For example, strawberry plants actually have 96 chromosomes. The composition of each of their chromosomes is also different than humans. So that just makes sense. Comparing human chromosomes to strawberry chromosomes. Human chromosomes need to encode for things like hair color, eye color, the fact that we have 2 eyes, 1 nose, 1 mouth, 2 arms, 10 fingers, 2 legs, 10 toes. Strawberry plants don't need any of that. They need to have things like genes that encode for carrying out photosynthesis, or creating roots, or shoots, or leaves, or flowers. They need to have a different set of instructions in their DNA than any other organism. Such differences in our DNA between different species, that's what encodes for variation between species, if we're going back to talk about what we learned in section 1. So there's variation between species, like why a human is different than a strawberry plant, because of the arrangement of those A's, T's, C's, and G's in our DNA. So we're going to go back now and we're going to watch that video about genetic mutations. We had talked about it a bit in the lab. So in the lab, I said your DNA is made out of that sequence of A's, T's, C's, and G's. Sometimes, though, something goes wrong when we copy our DNA to create a new cell. Sometimes those nitrogen bases can be removed and everything else kind of shifts upwards. And then all of a sudden your instructions in your DNA don't really make sense. Or sometimes an extra nitrogen base is added in, all the rest of them shift down, and then your instructions still don't make sense. So we're going to watch this video, learn about other types of genetic mutations, not just to do with the base pairs, but sometimes there's chromosomal mutations, which has to do with the chromosomes overall. And not every mutation is something detrimental. A lot of us just have mutations that don't affect us at all.
Turn on or turn off. Let's talk about sickle cell anemia. First, a little background. Hemoglobin is a protein with some really awesome ability. Or the Ninja Turtles. We loved the Ninja Turtles when we were little. We might be showing our age a little bit. But mutations really are not that glamorous. Most people understand that a mutation is some kind of change. It is a change of genetic material, and more specifically, a nucleic acid. RNA and DNA, they're both types of nucleic acids. So how does a change happen? Remember that in DNA, the base adenine A goes with the base thymine T, and the base cytosine C goes with the base guanine G. And that's all good, but what happens if the wrong base matches up? Many things can cause an error like that. There can be external factors, like chemicals and radiation, internal things too, like something goes wrong during DNA replication, during interphase. Interphase is a stage that prepares cells to divide during mitosis. And now while these things can increase the chance that a mutation is going to happen, it's important to understand that mutations, they're random. If a mutation is going to be a helpful thing for an organism, which is rare, very few mutations are actually helpful, it can't just will itself to get that mutation. An organism can't just will itself to get this. It's definitely not like X-Men either. And more about that when we get to natural selection. Many mutations are actually neutral in their effect, meaning they neither help nor harm an organism. And some mutations are harmful. So we're going to talk about the different kinds of mutations. First, we're going to talk about gene mutations. So DNA makes up genes, and genes code for proteins that influence or make up different traits. So when DNA has changes, otherwise known as a mutation, then different proteins can be produced, and this can affect an organism's traits. So let's look at the gene mutations. So first, substitution. That means you have the wrong base matched. So instead of A with T, you could put A with G, scandalous. You can have insertion, that means an extra base is added in. You can have deletion, that means a base is removed. Insertions and deletions have the potential to be especially dangerous. Because remember, in protein synthesis, we talked about how bases are read in threes. Well, if you add a base or remove a base, suddenly the number of bases total has changed, right? So, if you read in threes, depending on where it happened, everything that's read afterwards has the potential to get really messed up. We call this a frame shift. Now, these were all types of gene mutations, but we also have something called chromosome mutations. Remember that chromosomes are made up of DNA and protein. They're highly organized, and they have a lot of genes on them. So all the body cells in your body have 46 chromosomes. Human sperm and egg cells have 23 chromosomes. Well, changes can occur at that large chromosome scale too. So let's talk about these chromosome mutations. Just like insertion in a gene mutation, where bases got added, you can also have something called duplication in chromosomes. These are mutations where extra copies of genes are generated, so extra copies of that chromosome are generated. There's deletion, that's where some of the genetic material from the chromosome breaks off. Inversion, that's when a broken chromosome segment gets inversed, that means it gets reversed, and put back on the chromosome. Or translocation, we were not kidding back when we said there's a lot of trans words in biology. That's when a fragment from one chromosome breaks off and attaches to another chromosome. There's more mutations than what we just covered, but the idea here is there are many kinds of different changes that can happen. If a mutation is going to happen, there's also some vulnerable times, like when DNA replication happens during interphase, and also other times too, like meiosis. Remember, in humans, meiosis produces sperm and egg cells that have 23 chromosomes. But sometimes those chromosomes, when they're separating, they don't separate completely. We call this non-disjunction. This results in an egg or sperm that has too many or too few chromosomes, and that can cause a genetic disorder depending on which chromosome we're talking about. Different chromosomes contain different genes. So the specific chromosome that's affected, that really does make a difference in the result. And let's talk about some real-life examples now of mutations. Remember, we said that a lot of times mutations can have a neutral effect. Not all of your DNA codes for a direct protein. And other genes in your body can even influence whether those genes are going to be turned on or turned off. Let's talk about sickle cell anemia. First, a little background. Hemoglobin is a protein in your red blood cells that helps you carry oxygen. Well, in the disorder sickle cell anemia, the gene that codes for hemoglobin is mutated. And if you inherit 2 copies of this mutated gene, one from each parent, you can have this disorder. And the disorder makes it very difficult for your red blood cells to carry oxygen because the shape of the red blood cell, it's affected from this mutated hemoglobin protein, and this can lead to anemia and other problems. But get this, let's say you just inherit one copy of the mutated gene from one parent. You're going to be considered a carrier, but you don't really officially have the disease, and usually you don't even have symptoms. But what's interesting is carriers appear to have a protective factor against malaria. Malaria is a disease caused by a protist that can be transmitted by mosquitoes. These individuals, they can still get malaria, but usually their symptoms are less severe. So, in a way, this one copy of a mutation can be an advantage if you happen to live in an area where malaria is really present. Studying mutations and genetic disorders is a huge field right now. If this kind of thing interests you, there are a lot of careers to look into. In fact, there's a career known as a genetic counselor. They work to help families that may be at risk or affected by genetic disorders. They do a lot of good in the world. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so just to recap, DNA is made out of your nitrogen bases. There's 4 types. There's A, T, C, and G. A's always pair up with T, apples in the trees. C always pairs up with G, cars in the garage. When you put a bunch of nitrogen base pairs together, you get something called a gene. Genes encode for different traits like hair color or eye color. There's also different forms of a gene. Those are called alleles. Genes you get one set from your mom and one set from your dad. When you put a bunch of genes together, you get a chromosome. Humans have 46 chromosomes in all of our body cells, and just like genes, you get one set from your mom, one set from your dad. So each egg cell contains 23 chromosomes, and each sperm cell contains 23 chromosomes. When you put all of your chromosomes together, all 46, that's your entire set of DNA. So what I want you to do now is you're going to have a lined piece of paper, and you don't need to copy down the questions, but I do want you to try them. There's only 4 questions. Number 3 is especially a really good question. It's a higher level thinking question. So pause the video, do these questions, and when you're done, the answers will be on D2L.